Hello, welcome everyone. We'll get started here on this the afternoon workshop on population neuroscience and how to responsibly handle big data in the era of biobanks. And I think neuroimaging is moving in the direction of big data, and but some of the big data is also having an intersection with epidemiology. So I'm going to discuss what epidemiology is. So here's neuroscience, neuroimaging. We've got genetics, and what we're going to be focusing on during this afternoon session is really this intersection. But the principles that we're going to be talking about can be applied actually to every study. So what is epidemiology? And here's a definition I have from, from the Dictionary of Epidemiology. And it's the study of distribution and determinants of health-related states or events in specified populations, and the application of this study to the control of health problems. So I'm going to give an example of this, and this is a picture of a physician from the mid-1800s who was considered one of the fathers of epidemiology, and actually, to some extent, an imager, or a mapper at least, because what he did is a two-dimensional map of an area in London when there was an outbreak of cholera in the mid-1800s. And each of these dots here represent a death of someone, although at the time they didn't know what the cause of death was. So what John Snow did is he plotted the address of each of the people who died and looked at different pumps or wells where they got their water, and found that there was a relationship. So when we go back to the definition of epidemiology as the study of the distribution of determinants, what we see is the health-related states or events here are the deaths. They didn't know at the time that they were from cholera. The population here is in London, in Golden Square area of London. The distribution is the distribution of deaths here, or the distance from the pumps. And the determinants is drinking from the wells. And then the application is don't drink from pump A. So, and this applies also to current situations of diseases such as the Ebola outbreak, HIV and AIDS when that broke loose, and they didn't know the reason for things, and they called in the epidemiologist to figure out what was going on. So last year, at one of the keynote, Carla Miller presented this like a Manhattan plot of the UK-Biobank study, of which there were 2.8 million univariate cross-subject association tests between the statistically strongest associations of 2,501 imaging-derived phenotypes for different imaging modalities here, and 1,100 other variables in the UK-Biobank. So this has 2.8 million univariate analyses. So when I saw this, I'm thinking, okay, this is 2.8 million different tests. How are we going to deal with all this amounts of data in the context of false positives, true positives, false negatives, and true negatives, and just the nature, the magnitude of data? So within the Generation R study, we're also looking at this is a similar Manhattan plot, but this is different environmental and behavioral variables associated with autism spectrum disorders that were collected either prior to birth or at around the time of birth. And we also show actually large numbers, much more than what you would expect, that were significantly associated in this univariate analyses. And these are uncorrected as well. So one of the interesting aspects about environmental or health-related variables is that they're often correlated. So here's actually a connectogram that you might be familiar with is used in imaging, but this isn't imaging data. This is all the environmental variables that we have. And what you can see is that some are just highly intercorrelated with other variables. And these are different covariates we used here. But you can see that these are highly related. And this is different than genetic data, which if you're far enough away for language disequilibrium, then they're essentially independent tests. So, but environmental variables, also the ones that were shown in the UK-Biobank, are also highly correlated, which is going to inflate the number of significant tests. So, and this was described actually in terms of Smith and Nichols recently wrote a paper on statistical challenges in big data, and they gave, I thought, a nice example of this. For example, socioeconomic status might influence cheese consumption, and independently rate of cognitive decline in aging is reflected in the volume of the hippocampus. This would induce an apparent association between cheese and hippocampus volume that is not causal but induced by a common factor. If you look at cheese consumption without controlling for IQ, you might find a relationship. And actually, I was chatting with Tom Nichols, and he said a lot of this cheese, they're finding cheese related to a lot of different variables within the UK-Biobank study. So in 2005, John Ioannidis had a paper, Why Most Published Research Findings Are False. And so the goal of this workshop is actually to prevent this, so that we're doing high quality work that's replicated, and it's not going to be without bias, but we're going to talk about different techniques in order to help reduce those. So let's come back to this Miller paper, 2016, the Manhattan-like plot of environmental and lifestyle, many different variables here. And so one of the things that John Ioannidis said is that the number of false positives that you get is dependent on your number of hypotheses, but also the relationship between the number of true results versus the number of not true results within the hypotheses that are tested. So if we have 2.8 million hypotheses, let's say that 1 out of 10 is true, then we'd have 280,000 true findings. And the 1 out of 10 is the R that's used in the Ioannidis paper. But that actually is an unknown, really. We don't know how many true findings there are in all these 2.8 million. With 80% power, there'd be 224 true positives, and 56 with 80% power, that would be 56,000 false negatives. Alpha is less than 0.05, there'd be 126,000 false positives. And of course, 2,450,000 true negatives, which if you think about publishing true negatives, we'd have a lot of negative papers in this case, which probably wouldn't be a bad thing, but it'd be a lot of reading. Let's come back to, but that's in a perfect world. without bias. Because one of the other things that John Ioannidis did is he looked at, here's power, here's different types of studies, here's the R factor, so this is actually the relationship of the true positives to the hypothesis, to the non-true tests. So the best example here is a adequately powered randomized control trial with little bias, and so 1-to-1, so you expect a 50% chance of getting a true hit. And here you have the positive predictive value of about 0.85, which is quite good. What we're talking about in generally around the UK-Biobank is adequately powered. It's one of the largest studies that exists for imaging. This R is debatable. We don't know that, but he used 1 out of 10. The U is the bias, and we're going to be discussing different types of bias during the course of the afternoon. And you can see for adequately powered exploratory epidemiological studies, the positive predictive value drops here in this analysis to about 0.2. So, so this is one of the challenges that you see in terms of how do we do good, reproducible science. So there are many types of bias, and there's selection bias, there's reporting bias, analytic bias, systematic exclusion of certain individuals, there's attrition bias in longitudinal studies, there's funding bias, selection of important variables that favor the financial sponsor, and then career propagation bias, which is also called p-hacking. So with that, I'm going to talk about the schedule for this afternoon because it's a little bit different. That's in the printed version, but it's the same that's in the web. I mean, this is the online version, so it's so accurate in the online version, but it's different than what you see in the print. So next is Ryan, who's going to give an introduction to epidemiology. Anna-Marie Lauk is going to be talking about applied epidemiological concepts and actually a lot about confounding. Then we'll have a break, and then Amchi Kui will be giving a talk on, I think this is kind of self-explanatory. How do you deal with this massive amounts of data? And the talks are going to become increasingly technical during the course of the afternoon, so make sure you get lots of coffee here. And then Xinyan will be talking about replication beyond bash scripts and winner curses. So I will turn the mic over to Ryan.